Hello, everybody. Welcome to part two of the basic biology series. I'm going to continue on with some basic cell biology today, and you're going to learn some stuff today that actually is not normally in a introductory biology course, although I think it should be. But it's super important for understanding how cells work and how drugs that target abnormal cells work, which are the targeted chemotherapy drugs that we're going to be talking about. So if you'll recall from part one of this series, we went over this slide that you can see here, which is a very important process in the cell, probably the most important process that goes on in cells, which is the way that the information that's coded in the DNA, which is here in the nucleus, this double-stranded chemical, is copied onto a smaller chemical that's a chemical cousin of DNA called RNA. And then the RNA escapes the nucleus and goes to a structure called the ribosome, which is inside the cell but outside the nucleus. And then in the ribosome, the information on the RNA is used to put together proteins and other cell parts. Mostly proteins, though. Most RNAs have the instructions for making proteins. And human bodies require 20,000 or so proteins in order to function correctly, although different parts of the human body use different parts of those 20,000 instructions. So there's no cell in the human body, for example, that uses all 20,000 instructions because different tissues and different organs just use different subset of those 20,000. And actually that's what makes those cells different from each other, and what makes them look different, what makes them function different, is the fact that they're not making the same proteins in cell parts. There are some differences between them because they're using different parts of the DNA instruction. It's like if you give two chefs like the same recipe book, which has, say, 1,000 recipes in it, you can get very different meals because, one of the chefs may prefer the chicken and fish recipes, and they may look at those and use those. Another chef may prefer the steak and dessert recipes, and they look at those and use those. In fact, you can get entirely different restaurants where the chefs are all using the same recipe book, but they're using very different recipes from the book. For example, you can have a steak restaurant, essentially, and a dessert shop. And assuming you have a pretty comprehensive recipe book, which has steak recipes and dessert recipes and salad recipes and vegetable recipes and all that, you can have these completely different restaurants that are based on the same recipe book. It's just that the chefs in the different restaurants will be using, different recipes from the book. Like in the steak restaurant, they'll be using the steak recipes and the salad recipes and the vegetable recipes, and maybe, a couple of dessert recipes. But in the dessert shop, they'll be using mostly dessert recipes with maybe some sandwich recipes or salad recipes to go with it. So there might be a little overlap between recipes used in those two different restaurants, but mostly they'll be different ones because, you have a very comprehensive recipe book that has all these different recipes in it. And DNA is like that, scattered across the 23 or 24 different DNA molecules that you have in your cells are about 20,000 different recipes. That is, instructions for making 20,000 different proteins and cell parts. There's no cell in your body that makes them all. But every cell in your body uses some of them, some thousands of them. And, for a cell to function minimally, it requires, several thousand recipes to be active. And if you remember from the first video, the recipes, quote unquote, that are in the DNA recipe book are called genes. So each gene is the instruction for making one particular kind of protein or one particular kind of cell part. But as I said, cells actually require several thousand different genes in order to function correctly. And so what happens here is you have this process of taking the information that's on DNA and then ultimately using it to build a protein or a cell part by first separating the two DNA strands along a gene code, one gene. That's like opening a recipe book to one particular recipe, and then copying that recipe onto a little index card. So here we open the DNA molecule by separating the two strands along one gene code, and we copy that gene code onto a chemical cousin of DNA called RNA. And then we bring that single recipe that's on RNA out to the kitchen, which is our ribosome here. And then in the kitchen, we use that recipe to make our meal. The meal ultimately is our proteins here. And proteins, just like meals are made of ingredients that you put together in particular ways, proteins are made of smaller chemicals called amino acids, which are linked together. They're represented by these balls here, these colored balls. And just like, meals are made of ingredients, or words are made of letters that have to be put together in particular order so the word makes sense. Proteins are made of these amino acids that have to be put together in a proper order so that the protein will function properly, will do its job. And the job of all proteins is to attach themselves to other chemicals and to do things with those chemicals. Each different protein that you make is designed to attach itself to just a few specific chemicals and do things with those chemicals. The main chemical that a protein attaches itself to that it's supposed to do something with is called the substrate chemical for the protein. And different types of proteins have different substrates. But in order for a protein to attach itself to the right substrate, it has to be made in the right way, which means the right order and number of amino acids. And that information is coded on the messenger RNA. And then in the ribosome, it's decoded, essentially. And the information on the messenger RNA is used to put together proteins with a protein with the right order and number of amino acids so that the protein folds up and works correctly. So if a cell, for example, needs to use, let's say, 5,000 different genes in order to function right, then it's got to be able to open up the DNA at each of those 5,000 genes to separate the strands to build messenger RNAs from those 5,000 genes, and then to bring those messenger RNAs to the ribosome where they can be used to order up amino acids, put amino acids together in the right order, so that you make those 5,000 different proteins that perform their functions correctly. Okay, so that's the basic process of turning DNA information into, working cell parts like proteins. These are the working or business parts of the cell, the parts of the cell that actually do things that the cell needs doing. Okay, so last time we talked about cancer as a mutational disease, and we'll get back to that. But before we go to that this time, I want to sort of go backwards a little bit and talk to you about how cells normally function. Cancer is an abnormal functioning of cells. But let's talk about how cells normally function. First of all, you've got different types of cells in your body. And, the basic question that biologists had for a long time is, if all the cells in our body have the same DNA information, same genes with the same codes, same instructions, then how is it that we can have, from that same set of information, we can have a liver cell which looks and functions different than a brain cell, which looks and functions different than a muscle cell, which looks and functions different than a skin cell. We can make all those different types of cells from the same instruction set. So how do we do that? Well, just like we could open different restaurants, using recipes from the same recipe book. Different types of cells in your body transcribe different parts of the DNA, different genes, and then translate those different genes into proteins or other working cell parts. So for example, we might be building a protein here that's used in liver cells, but not in kidney cells. In kidney cells, this part of the DNA would not be transcribed. That is, you'd never open the DNA here to copy the DNA code into RNA. That wouldn't happen in a cell that didn't use this gene. And some of your cells don't use that gene. Maybe your kidney cells don't use that gene, but your liver cells do. And so the liver cells can open up the gene here, separate the two DNA strands, build the RNA copy so that particular protein can be made. Whereas in your kidney cells, this part of the DNA never gets opened. You might think, well, how is that done? And we're going to go over that in a minute because it's super important. Even once the protein's made, your cells have to be responsive to their environment. One of the characteristics of living things, which cells are, is it's something that can sense its environment and respond to it. It's like you use your senses to see what's going on around you, or hear what's going on around you, or feel what's going on around you. And you can respond to the things that you see, the things that you hear, the things that you feel. That's the whole point, is so that you can respond to what's going on around you. You can change what you do in response to what's going on around you. And that's just a very basic, the ability to sense and respond to the environment is a very basic characteristic of life. But it requires that living things be able to change what they're doing. And usually to change what they're doing pretty quickly. So you have, on the one hand, the ability of different types of cells to use different parts of the DNA. That's what ultimately makes them into different types of cells. And then you have the ability of cells, even within the same tissue, to sense and respond to changes in their environment, and to do that fairly rapidly. So the first kind of response, when a cell decides what kind of cell it's going to be, and which parts of the DNA, therefore, it's going to use and which part it isn't going to use, that's under what's called epigenetic control. That's control that involves transcription at the DNA and RNA level. That's really what determines which genes get used and, what proteins get made in the cell, and so what the cell's ultimately going to become. But then when any given cell needs to respond to rapid changes in the environment, then you get what's called post-translational modification of proteins, which allows you to change the shape and function of a protein in a very quick way, and usually in a temporary and reversible way. So, you can take a protein that's more active and make it less active, or one that's less active and make it more active. You can do that pretty fast. Now, I want to go back to an analogy here because analogies are helpful. Let's take chefs that are opening two different restaurants. Again, they're using the same recipe book, but one chef wants to open that steak restaurant and the other chef wants to open a dessert restaurant. So salad and desserts in one restaurant, and steak and potatoes and vegetables and in the other restaurant. So you can do that even with the same recipe book, because the recipe book has recipes for steak, but also for potatoes and other vegetables, and for salads and for desserts. So you can open both of those restaurants just by using different recipes from the same book. So, the dessert restaurant's never going to use the steak recipe, so it's going to keep those pages closed where the steak recipes are. And the steak restaurant is, not going to be making most of those desserts, so it's going to keep most of the dessert recipes closed up. But there's also fine tuning that even once you open your restaurant. So now I know what kind of restaurant I've got. I've got my dessert restaurant and I'm opening my dessert restaurant. But based on customer feedback after I've opened my restaurant and, maybe what kind of crowd I've got that day, is it a weekend? Is it a weekday? Is it lunch? Is it dinner time? I may adjust my menu a bit, right? Maybe I always make the same desserts, but for example, I find that some desserts are more popular in the evening and some desserts are more popular in the afternoon. So, when the evening rolls around, I'll make less of these desserts and more of those desserts. And, in the afternoon, I may reverse that, make more of that original dessert and less of something else. And also based on customer feedback, I may find that, for example, my customers they love my lava cake, but they don't particularly like my cinnamon muffins. And so I may adjust the menu again so that, every day I actually make more lava cake than I did originally, but I make fewer cinnamon muffins. I don't completely take them off the menu, I just make less of them. And so that would be the equivalent of post-translational control in a cell, based on the cell's experience and what it needs at the moment and what part of its life cycle it's in, etc. It may need certain proteins to be more active and certain proteins to be less active. Even the less active ones, it may still need some activity from them, but just not as much, depending on environmental conditions. So these post-translational modifications of the proteins themselves are able to sort of act like a dimmer switch on a light. And rather than just turn the light on and off, you can make it brighter or dimmer, essentially more active or less active. And that's what's called post-translational control of cell function. So you've got post-translational control, which is by modifying the proteins, and we'll talk about how that's done, and then to make them more or less active. And then we've got epigenetic control at the level of the DNA and RNA, which is really about, which genes we're going to use and which genes we're not going to use. The epigenetic control is more about, determining what type of cell a cell is going to become. And then the post-translational control is that cell's ability to respond to changes in its environment by, increasing certain functions and decreasing others, by making certain proteins more active and certain proteins less active. Okay, so again, this is all normal. This is not abnormal. Okay, we talked last time a little bit about cancer as an abnormality of cell function. This is actually normal cell function. So let me give this to you in words. I'm going to take this away. And I have to make myself very small here. Go off in words. So how cells regulate their gene and protein function. You've got liver cells, you've got kidney cells, they look different, they behave differently, they have different functions. So how does that happen? Because, they both have the same DNA with the same information. And this has mostly to do with which genes are activated and which genes are not in these cells. So again, there is no cell that activates every gene that it's got. All cells have more genes than they're actually going to use at any given time. So what makes the liver cells different than the kidney cells is what's called epigenetic control, which is the ability of the cell to use receptor proteins to help them determine, based on their location and what's around them, which type of cell they should be. Should they be a kidney cell? Should they be a liver cell? And then to control transcription based on that determination. So if I determine based on my location, based on the cells around me, if I determine I should be a liver cell, then I'm going to turn off most of the kidney specific genes. I'm going to make sure that those genes don't get transcribed, that part of the DNA doesn't get separated. But I'm going to turn on the liver specific genes. I'm going to make sure that those are active, that those can be separated, the strands of DNA there can be separated, and those genes transcribed into RNA, which can then be used to make proteins. So there's 3 different levels of epigenetic control. And let's see, I'm going to show you one of them. This is the big one up first. Now, DNA, when it's in the nucleus, is in structures called chromosomes. They look like little X's. You've probably seen pictures of that. And what chromosomes actually are is they're the 2 DNA strands, this is the DNA, wrapped around proteins called histone proteins, histone. Histone. So the DNA, the parts of the DNA that are wrapped tightly around these histone proteins, sort of like thread around a spool, they cannot be, the genes in these parts of the DNA cannot be used, cannot be transcribed, because when the DNA is bound tightly to histones, the 2 DNA strands cannot be pulled apart. So these genes, as it says here, are inaccessible. The DNA is turned off there. And those genes are not going to be used by the cell. But then there's other parts of the DNA where it's looser, where there's a lot of DNA that's just loose and free and not wrapped around the histones. And those parts of DNA contain genes that are accessible, that can be used. So the first bit of epigenetic control is in which genes, which parts of the DNA are tightly bound to the histone proteins in the nucleus, and which are loose and sort of float between the histone proteins. Now you could ask what determines that, and the answer is chemical modifications of the DNA and the histone proteins. So the cell can, based on signals from outside the cell has proteins that go to certain parts of the DNA and certain parts of the histones, and adds some small chemicals to them that make the DNA very attracted to the histones and make it stick really tightly to the histones so it can't be pulled off or unwound. And then other parts of the DNA are modified so that they won't bond to the histone proteins. And that's the DNA that's free and in between histones that can be used for transcription. So there are protein receptors in the cell that get signals from the external environment, which directs them to go to certain parts of the histones and certain parts of the DNA and modify them so that the DNA there is shut off. It wraps tightly around the histones and is shut off. And then go to other parts of the DNA and modify the DNA so that it won't bond to histones. And those are the parts of the DNA that are used. Different chemical signals from outside the cell will lead to different parts of the DNA being wrapped tightly or unwrapped. And this tends to be a fairly permanent condition for the life of the cell. That is, which parts of the DNA are tightly bound to proteins and which aren't. It's hard to reverse this kind of interaction. Not impossible, but difficult. And so this is usually the first set of epigenetic controls that leads a cell to become either a liver cell or a kidney cell, for example, is this bonding, how tightly the DNA is bound in certain areas to histone proteins in the nucleus. And let's see, let's go back here. Okay, so that control is up here in the transcription part. The parts of the DNA that are tightly bound to histone proteins, you can't separate the DNA at those parts. And so you can't do transcription there, which means you can't make the messenger RNA copy of that gene, which means you can't turn that gene into a protein. Now there's other kinds of epigenetic controls at the transcription level. For example, there's a protein that pulls the two DNA strands apart at a gene. Starts at the beginning of the gene and pulls the DNA strands apart all the way to the end of the gene. And that protein is called RNA polymerase protein. That's the primary transcription protein. That particular protein can be directed to or away from certain genes by other chemicals that are called activators or promoters or repressors. So if there's, for example, a promoter chemical that directs the polymerase to the beginning of this particular gene so that it can start transcribing this gene. But in other cells, there may be a repressor active that prevents the polymerase from opening up this gene. The difference really between the control with histones and the control with RNA polymerase is with histones, you're actually inactivating entire sections of DNA, which contain many, many genes. So, you might be inactivating 50 or 100 genes at a time using the DNA histone association. But with these polymerase activators or blockers, which are called promoters or repressors, there your control is finer. You can keep one gene active while where you can inactivate the next gene over from it. So instead of inactivating whole groups of genes, you can just inactivate like one gene that's sitting between two active genes, for example. So it's a finer level of epigenetic control based on which repressors and which promoters are active in the cell. Also, that's a more reversible interaction usually than the histone interaction, which tends to be pretty irreversible. There's another kind of interaction, which we only discovered fairly recently, which is epigenetic. And that's that in some cells, the cell can make RNA from a gene, but it also makes anti-RNA from a different gene. So there's another gene that creates an anti-RNA that can bond to this RNA. And when you've got those two RNAs bound to each other, you form a double-stranded RNA, which the cell usually destroys pretty quickly and also is too big to get out of the nucleus and into a ribosome. So you might have certain genes active and they're actually getting transcribed, but then you're also activating genes that are anti-transcription for those genes that make RNAs that bond to those RNAs that you make from those genes and prevent them from being translated. So that's another form of epigenetic control because it involves genes and RNA and DNA. And again, that's a finer level of control than the histone control, where you inactivate or activate, large sections of DNA and large numbers of genes at the same time. Here, you're working really with just, this gene versus that gene, one gene next to another one, where, one might be active and the one next to it is inactivated, either because of a repressor for DNA polymerase or because of an anti-mRNA that's made from a different gene that blocks the translation of one of those RNAs but not another one. Okay, so those are your epigenetic control levels. But again, sometimes you actually do need the activity of a protein, but you also need the ability to make that protein more or less active, depending on the environmental conditions of the cell, which might be changing rapidly. Like, for example, there's a lot of genes that are turned up or down based on whether you've recently eaten a meal. That is, they're activated or deactivated by changes in your blood sugar. And your blood sugar changes throughout the day. In a single day, your blood sugar is going to go up and down quite a bit. And in response to that, certain genes in certain cells are activated and their activity is increased, and other genes, their activity is decreased. For example, after you've eaten a meal, the activity of proteins that import sugar into the cell is increased. the activity of proteins that bond sugar molecules together to make starch is increased. The activity of proteins that take apart starch molecules is decreased. None of those proteins are completely inactivated, but their levels are made substantially faster or slower depending on how much blood sugar there is. And during the day, after a meal, when your blood sugar goes up, you get modifications of those proteins. But then when you've been sleeping for hours and your blood sugar goes back down, then you get the reverse of that activation. So all of that is based on, most of that is based on post-translational activation, which is very rapid. This kind of activation can occur within seconds or minutes of an environmental change. And it tends to be completely reversible. That is, you can make a protein more active and then make it less active again. For every post-translational control you have of a protein that makes it more active, there's generally another control in that same cell which can also make it less active. And which of those systems is more vigorous at any given moment determines whether your protein is more active or less active. And again, that can change, fairly rapidly throughout the day. And so you can reversibly change the activity of protein, make it more active during certain parts of the day and less active during others. Okay, so let's go back to the words here. We've got epigenetic control of transcription, that is control of the DNA histone level, at the RNA polymerase level, at the RNA messenger RNA level. This kind of control, which tends to be longer term control, is used to create different cell types when your body's developing and when you're still inside your mom, basically. So this kind of control is used to determine whether your cell becomes a cell of this organ or that organ. Epigenetic control is also used to repair organ damage because it can be used to turn what are called adult stem cells, which are cells left over from when you were an embryo, which never got turned into an adult tissue. Your body keeps some of these embryonic stem cells around in adulthood so that it can more easily repair damage to an organ, because those adult stem cells can then be turned into a regular cell, like a liver cell, through epigenetic control and changes in epigenetic control. So stem cells are used to repair damage to organs in older age. And that's based primarily on epigenetic control of the genes that can turn a stem cell into that organ. And then the processes involved with changing stages of life, like when you go from being an adolescent to an adult, or when you reach old age, and you get the processes involved with, cellular aging, those also tend to be epigenetic. Also, the processes involved in cell death. Under certain conditions, your cells are programmed to activate certain genes, which are like suicide genes, where they kill themselves, essentially. They activate genes that are lethal to the cell. And they do it deliberately if the cell is behaving abnormally. So that programmed cell death, program means there are actual genes for it that cause it. And some of the changes in the cell involved in programmed cell death are actually epigenetic changes. Okay, so again, you have the DNA packing and unpacking, which is how tightly it's bound to histone proteins, which can tie up entire sections of DNA with many, many genes in them. That's the primary method of epigenetic control, but there's also transcriptional control when the RNA polymerase protein bonds with promoters. The promoters can guide that protein to the beginning of certain genes, so you can transcribe those genes. And then there are repressors, which will which will prevent the RNA polymerase from getting to the beginning of other genes, so it can't transcribe those genes. And then you've got these small interfering RNAs where there are certain genes in your genome which can make RNAs which bond to the messenger RNA of other genes. And when these RNAs are bound to messenger RNA, they lock up the messenger RNA so it can't be used for translation or turned into a protein. And then there's, excuse me, then there's the post-translational control of protein activity. Now, how can you, and this is essentially, you need to respond to a quick change in the environment of the cell, not by creating whole new proteins, but just by taking the proteins you already have and making some of them more active and some of them less active. That's post-translational control of protein activity. And that can be done at the protein itself, whichever one you want to turn up or down. Most proteins, in addition to having a binding site for their primary substrate, the chemical they're supposed to grab onto and do something with, most proteins also have regulatory sites on them, which are can bond to activators, chemical activators, or chemical inhibitors. So most of these, most proteins have an active site which they use to bond to a substrate, and they have regulatory sites that they use to bond to inhibitors or activators, which are smaller chemicals usually that cause the protein to become more active or less active. Another method of control, again, this is more recent discovery, another way to control how active a protein is by controlling how long the protein lasts in the cell. That is, after the protein's made, does it last a long time in the cell? I mean, is it there for hours or days doing its thing? Or does the protein itself break down in a very short period of time, like maybe a few minutes? If the protein breaks down very quickly, then its activity doesn't last very long. And that will reduce its activity unless you keep making more of that protein constantly. So one way to control protein activity is by controlling how long the protein lasts. And some RNAs are messenger RNAs are more stable than others naturally. The ones with more stable messenger RNA, the protein tends to last much longer because you can keep making it over and over again. But there's also ways of targeting proteins for destruction. You can put these essentially molecular bullseyes on proteins. One of them is called ubiquitin. That's a molecular bullseye. And when ubiquitin is added to a protein, that targets the protein for destruction in an organelle called a proteasome. Proteasomes are organelles with digestive chemicals that break down proteins. So if I've got a protein that I want to control by making it not last very long in the cell, then I will add a bunch of ubiquitin molecules to that protein, and that will target that protein to the proteasome. It will make it rapidly go into a proteasome where it gets chopped up and destroyed. So that dramatically lowers its natural lifespan, and it becomes less active overall as a result. Now, there are proteins that are just floating free in the cell fluid. They're called cytosolic proteins. Those proteins are destroyed in the proteasomes. But then there are other proteins that are in like the cell membrane or in various organelles of the cells. Most organelles of cells have their own membranes, like lysosomes and, the plasma membrane and mitochondria, etc. And so proteins that are natural part of organelles are destroyed in the lysosomes by a process called autophagy. That means phagy, by the way, means eat in Latin. Auto means either automatic or self. So think of autophagy as self-eating. And lysosomes essentially digest organelles that have a lot of old proteins in them and aren't working very well anymore. Rather than try to keep those organelles alive when they're malfunctioning, the cell will just digest the proteins in them and destroy the organelle. There's an ERAD system, which is specifically to just destroy proteins that are in an organelle called the endoplasmic reticulum. That's the ER in ERAD. This is how you can tell a biologist from a normal person. Normal people, when they hear ER, they think emergency room for a hospital. Biologists, when they hear ER, they think endoplasmic reticulum, which is an organelle in all eukaryotic cells. And it has its own protein destruction system, which is different than these other two. But in any case, determining how long a protein lasts in a cell by determining how quickly it gets broken down is a primary control method that cells use to keep their bad proteins from getting out of control. And one thing we've discovered recently is that there are a lot of diseases, especially neurological diseases, that occur because bad proteins are allowed to accumulate in cells without getting broken down. There's essentially a defect in the ability to break down certain proteins, to target them for breakdown and then to break them down in these proteasomal structures or lysosomal structures, or ERAD structures. And if this happens in the brain, if proteins that are no longer working right are allowed to accumulate in brain cells, they become toxic to the brain cells. They can destroy the brain cells. And then you get chronic brain diseases from that. Things like Alzheimer's, Parkinson's, ALS. These are diseases where abnormal proteins build up in brain cells because they're not being destroyed properly. They're not being targeted for destruction, and the organelle parts that normally destroy them aren't working right. So this is a pretty important concept in biology, the concept of protein stability by controlling the timing of protein breakdown. By the way, in cells, when proteins break down, we call that degradation or destruction of proteins. So, for example, in Alzheimer's, a couple of different types of protein, amyloid and tau, build up and badly affect the cells of the brain because they are not being properly targeted for destruction. Those proteins, which become abnormal, they should be broken down so that they can't hurt the cell. But in the case of Alzheimer's, they're not broken down, and so they do hurt the cells of the brain. Okay. So, very important here is that in order for cells to function right, they have to be able to silence certain genes and activate other genes depending on which type of cell they are. They'll be silencing and activating different sets of genes. But they also had to respond properly to their environment. They have to be able to post-translationally control protein activities, either by controlling how long the protein lasts in the cell before it's destroyed, or by determining protein activity by bonding inhibitors or activators to regulatory sites on proteins to make them more or less active. And I'm going to show you pictures of this in a minute. Now, what has all this got to do with cancer? That's a good question. It turns out that cancers are not the result of normal regulation. All of this is normal cell regulation. You don't get cancer from normal cell regulation. It's normal for cells to silence certain genes and activate others. It's normal for cells to modify certain proteins to make them more active and modify other proteins to make them less active, and then reverse that when conditions change. So all of this is normal functioning of the cell. What's not normal is to change the instructions for a protein. Okay, remember the instructions for the protein are actually laid out in the order of chemicals that are creating the DNA molecule. DNA is a big molecule which is made by bonding together smaller molecules called nucleotides. And there's 4 different kinds of nucleotides, which we usually abbreviate A, C, T, and G, that are in a particular order along the DNA. And the order of these nucleotides in a gene is what's copied onto RNA, which has similar nucleotides, and then what's used in the ribosome to create a protein. So what's not normal? What's normal is for the cell to control which genes get activated. That is, where in the DNA you can separate the 2 DNA strands and where you can't. Which proteins get active, which proteins get modified so they're more active, which proteins get modified so they're less active. All of that is normal. Not normal is to change the actual instructions for making a protein. Because to change the instructions for building a protein correctly, that involves what we call mutation, which is a change in the order of nucleotides in the DNA. So for example, if this part of the DNA, you have a G nucleotide followed by a C, followed by an A and a T and a C and an A and a C and an A. If you were to change, for example, this second C here to a T nucleotide, then the code, instead of reading GCATC, it reads GCATT. Okay, well, that's a change in the code, in the instructions. And changing the instructions for building the protein actually changes 1 or more amino acids that are in a certain position in the protein. For example, this change here might change this particular amino acid into an entirely different amino acid. And that's going to change the order of amino acids in the protein, which is going to change the function of the protein. Usually it'll make it less active or maybe not active at all, because proteins often have to be made exactly right to work exactly right. But there's different kinds of, there's mutations in different parts of proteins. For example, you get a mutation in the active site of a protein where amino acids in the active site are changed. That usually leads to the protein not working, or not working very well because it can't bond to its normal substrate. But you can also get mutations in different parts of the protein. For example, you can get a mutation in a regulatory part of the protein, which bonds to an inhibitor. And in that case, the protein doesn't become less active, it actually becomes more active, because it can no longer bond to the inhibitor that normally cools down that protein, makes it less active. Since it can't bond to that inhibitor anymore, the protein is always active, even when it shouldn't be. And so that mutation in a regulatory part of the protein can sometimes, depending on which regulatory part it's in, can sometimes make a protein less active, but sometimes it makes it more active. And of course, mutations in promoter regions, in promoters or in repressors, can make it so that you can more easily or less easily transcribe certain genes, so that more of that protein is made or less of that protein is made, depending on which way the transcription goes. Okay, so mutations, which are not normal, changing the actual DNA code for building a protein so that the protein is made differently, That's not normal. That's like in the analogy of a recipe book. That's changing the recipe. That's substituting substituting cornmeal for flour or sugar for salt. Okay, that's a real change in the recipe. That's going to dramatically change the food that you're making, the meal that you're making. And so that's what mutation does. But because mutations can be in regulatory genes or regulatory parts of proteins, in addition to the main parts, they can have various effects on the cell. They can make certain proteins more active, certain proteins less active, certain proteins completely inactive. They can cause you to not transcribe a protein you normally do transcribe, so you lose the function of that protein. But they can also, if, for example, you get a mutation in a repressor, which allows then polymerase to activate a gene which isn't normally active, you can get a function in a cell from mutation that normally isn't in that cell. Because the cell's now making a protein that it doesn't normally make. It's one of the proteins that DNA has the instructions for, but it's not normally made. So depending on which part of this system, this control system is mutated, you can get, in some cases, proteins that are more active, in some cases, proteins that are less active than normal. In some cases, you can get proteins not made that should be made in the cell, and in some cases, you can get proteins that are not normally made by that cell to get made. Okay, and all those things will dramatically change the functioning of the cell. So when we talk about cancer, we're talking about mutations in critical genes that in some cases make the genes more active and in some cases less. Now, why is that important? Well, because modern chemotherapy, what's called targeted chemotherapy, is based on our understanding of which genes are mutated and in what ways. So for example, because we're trying to specifically target the mutations that are causing that cell to become cancerous. So for example, if we have a mutation in a protein that makes it less active, then a targeted therapy would be to make that protein, to try to make that protein more active again. If we have a mutation in a protein that makes it more active than normal, then the treatment would be to make that protein less active. Now, your DNA, because cancer has been with us as long as there's been an us, and I think we talked about this last time, cancer is not a new disease, it's an ancient disease. And it's been with humans probably as long as there have been humans. Some of your genes are designed to prevent your cells from becoming cancerous. So you actually have built-in cancer protection in your genome, in your set of genes. The genes that protect you against cancer are like DNA repair genes, which can fix mutations. Remember, cancer is a mutational disease when you get mutations in critical genes, but you have proteins in your cells that are designed to detect mutations and to fix them before they're passed on to new generations of cells. And those are essentially anti-cancer genes. So DNA repair genes are anti-cancer genes. If your DNA repair genes get mutated so that they don't work or don't work as well, that's a mutation that encourages cancer. That's an oncogenic mutation. So in that case, you're taking an anti-cancer gene and making it less active. So when you make an anti-cancer gene less active, that means you're more likely to get cancer. So let's go down to targeted cancer therapy here. Okay. So one way to to do specific targeted treatments for cancer is by targeting oncogenes. But to do that, you have to know, which genes are oncogenes, which oncogenes have been altered in these particular cancer cells, and what's the best way to reverse that effect. So we go down here to this part, this lower part here. Oncogenes are the genes that, when they're mutated in critical, can make a cell cancerous if enough of them get mutated. And these oncogenes tend to be either DNA repair genes or cell reproduction genes or cell suicide genes. Generally, DNA repair is anti-cancer. Those are anti-cancer genes because they can fix mutations that lead to cancer. So when you have anti-cancer genes, the mutations that give you cancer are the ones that make those less active. Like the ones that make DNA repair proteins less active. That's an anti-cancer mutation, an oncogenic mutation. Sorry, that's a pro-cancer mutation because the anti-cancer gene is less active. Also, the genes that promote cell suicide. Cell suicide is when cells activate certain genes that kill themselves. So these genes make proteins that just chop up the insides of the cancer cell or prevent the cancer cell from reproducing. Cell suicide genes are also anti-cancer genes because when a cell is becoming cancerous, it starts behaving abnormally and functioning abnormally, and the suicide genes are normally activated in those conditions. When cells are behaving abnormally, that activates the suicide genes. So a oncogenic mutation would be one that deactivates the suicide genes, because these are genes also that protect your cells against cancer. So instead of becoming cancerous, they destroy themselves. Or if they picked up a potentially cancerous mutation, they fix the mutation before it has a chance to wreck the cell. So DNA repair genes, cell suicide genes, those are anti-cancer genes. When those get mutated so they work less well or don't work at all, that's an oncogenic mutation. Now, cell reproduction is the opposite. Cell reproduction is actually a pro-cancer gene. That is, if you made the cell reproduction gene products much more active, you'd get cells that reproduce too quickly and can form tumors. So making these genes more active can make the cell turn cancerous. Whereas making the suicide genes or the DNA repair genes more active makes the cell less cancerous or less likely to turn into cancer. So in the case of the cell reproduction genes, there's a lot of cell reproduction genes, and pretty much all of them, if they're mutated to make them more active, that can give you cancer or can lead to cancer ultimately. And so the treatment for that is you want to make those genes less active. Nowadays, we can do what's called oncotyping of tumor cells, which gives us an idea of which of these oncogenes are mutated in that particular cell line, and then in what way. Are there suicide genes that are mutated to make the cell less able to commit suicide? Are there repair genes make the cell less able to repair its DNA? Are there reproduction genes that are mutated so that they become more active and make the cell more likely to reproduce too quickly and form tumors? Those are all oncogenes. Because we can do genotyping of cancer cells, we can figure out in many cancer cells which of these oncogenes are overactive, which are underactive. And for some of these, we already have treatments which can reverse that effect. So that would be a targeted treatment for that particular kind of cancer. Okay, questions about this? I give a couple of specific examples here. For example, a gene called the RAS gene produces a protein that makes a cell much more likely to reproduce and to grow faster. And one particular mutation in the RAS gene called KRAS mutation produces a protein, abnormal RAS protein that's always on. Essentially, it can't be turned off or inhibited. And so the cell with that KRAS mutation becomes more reproductive. Its reproduction is accelerated. And that can lead to it forming a ball of extra cells, which we call a tumor. So the treatment for that is a chemical treatment which sticks to the RAS protein, actually sticks to the KRAS protein, and makes it less active. So that slows down the reproduction of cells, the growth of the tumor. But then another gene that's commonly mutated, by the way, RAS is mutated to KRAS in about 30% of all cancers. So about 30% of all cancers, the cells have a KRAS mutation, which means treating this mutation, really can help treat a lot of different cancers. An even more common mutation in cancers is what's called a p53 mutation. P53 is an anti-cancer cell suicide gene. So if you get a mutation in p53 that makes it less active, then cells become unable to commit suicide, even when they're behaving badly and they're very disordered internally. The cell can't commit suicide. And so when the p53 gene gets a mutation that makes it less active, that makes the cell more likely to become cancerous. When RAS gets a mutation that makes it more active, that makes the cell more likely to become cancerous. So the RAS oncogene is a mutation that stimulates the RAS protein and makes it more active. The p53 oncogene is a mutation that makes p53 less active. And so the cell is less good at killing itself, basically, when it's disordered inside. Unfortunately, the changes in the anti-cancer genes that make them less active are harder to fix than the mutations in the growth genes that make them more active. When you have like a broken anti-cancer gene, the fix is difficult because you have to make that more active. But the problem is it's broken to make it less active. So making it more active is pretty difficult. Whereas genes that normally increase the probability of getting cancer when they're more active are easier to treat because all you have to do is find something that inhibits their activity. And finding a chemical that inhibits the activity of a protein that brings the activity down is usually a lot easier than finding a protein that builds up the activity. That's a trickier thing. So oncogenic cancer therapy is generally easier and more effective when it's targeting a mutated pro-cancer gene than when it's targeting a mutated anti-cancer gene. Okay, so that's quite a lot, but that's the reason that I was giving you some of the normal regulation information, is because mutations in the normal regulatory pathways for cells can cause quite serious disorders of the cell, including leading to cancer ultimately. So I want to do some illustrations and some modeling here now. Let's start with some illustrations. Okay, so here's a protein. And when this protein is active, it opens up what's called its active site, which is where the normal substrate of the protein bonds to the protein, so that the protein can do something with it. different proteins do different things with their substrates. For example, let's suppose this protein breaks its substrate apart. When the substrate bonds to its active site, then it can break the substrate apart, and that's what the protein's supposed to do. It's supposed to grab onto a chemical and break it. Okay, but it can only do that when the active site is open. Now, this protein also has regulatory sites. It has an inhibitor regulatory site here, and it has an activator regulatory site here. So when there's an inhibitor chemical bonded to this inhibitor site on the protein, that means the protein never opens up its active site. The active site is never exposed. And so the protein is essentially inactivated when there's an inhibitor chemical there. But when there's no inhibitor chemical there, and when there's an activator chemical around, the activator chemical bonds to the activator regulatory site of the protein, and that keeps the active site open, which allows the protein to bond to its normal substrate and break it apart, which is what it's supposed to do. So when this protein is bonded to an activator, which bonds to the activator site of the protein, then the protein does its normal function. When this protein is bonded to an inhibitor, then its activator site, its active site is closed, and it can't do its normal function. Now again, mutations can mess with this regulation. If there's, for example, let's say there's a mutation in the inhibitor site of the protein, so that the inhibitor site cannot bond to the inhibitor chemical. Let's say the site just completely disappears. Then this protein cannot be shut off. Okay, once it's activated with the activator, then it's on always, and it can't be shut off. So this protein then, without this site down here, will stay mostly in an active state where it's sort of constantly doing its job. This would be the equivalent of a KRAS mutation. With KRAS, the inhibitor part of the site on the RAS protein is destroyed by the mutation, so that the protein cannot bond its inhibitor, which means it can only bond its activator, which means it's pretty much always active. Now that's bad if you're a growth protein and a reproductive protein, because then the protein just keeps reproducing too fast, forms a tumor. Okay, so this is sort of an example of the activator and inhibitor sites. This is post-translational control of a protein activity, but we get out of control when we have mutations that destroy one of these sites. Mutations can destroy the active site. Other mutations can destroy the activator site. Both of those would cause the protein to be primarily inactive. But you can also get mutations which destroy the inhibitor site, which would make the protein pretty much constantly active or overactive. So this work was verified by a number of different tests and experiments, but I'm going to show you a primary one. And I'll have to enlarge myself here so you can see me better. I feel sorry for you, actually, but here we go. So let's suppose this is a protein. I'm going to use tinfoil here to model a protein. If the protein's made correctly, it's going to take its proper shape, which is this. Here's its proper shape. And with the proper shape, it has an active site here. And the active site can bond to the normal substrate of the protein, which, let's say that's starch. This is a protein that breaks apart starch molecules. To do that, it has to bond to the starch molecule in its active site. So here's the starch molecule, here's the protein, here's the active site of the protein that bonds to the starch. You can see it's got the right shape to fit that starch molecule so that the protein can pull the starch molecule apart. But this protein also has regulatory sites. It has an activator site here, and it has an inhibitor site here. And then here's the active site here. So normally, if there's an activator chemical which is bonded to the active site of the protein, that opens up, I'm sorry, if there's an activator which is bonded to the activator site, regulatory site, that opens up the active site of the protein so that the protein can bond to starch. So when the activator is bound to the activation site of the protein, the active site is open, and then the protein can bond to the starch active substrate and take it apart. But if the protein is bound to the repressor chemical, here's the repressor chemical. So the repressor chemical here bonds to the repressor part of the protein, and if that's bonded in, then the active site closes up. So now we have a protein with a closed active site which can't bond to its substrate. Okay, but all of this is reversible. So when this inhibitor comes off, and when the activator comes back on to the active site, then the protein opens up again, and then it can bond to starch again. Okay, so the key to these protein modifications is that they're all reversible. And it depends on the conditions. If there's more activator around than there is inhibitor, then the protein's going to be more active. If there's more inhibitor around than there is activator, then the protein's going to be less active. But mutations can really mess up the system, because if the mutation is in the part of the protein that becomes the active site may not be made correctly. It may be made like this so that the protein can never bond to starch. Then it becomes a completely and permanently inactive protein, and that's not reversible because that change was not in the protein, it was actually in the DNA. It was in the instructions for building this protein. So the protein was built wrong because one of the amino acids in the active site was incorrect. So now the active site is all closed up, and the protein can't bond starch, so it can't do its job, which is pulling starch apart. But what if the mutation was in one of these regulator sites? So here's the inhibitor site. If the mutation was in the inhibitor site, it may have closed up the inhibitor site. But with the inhibitor site closed, now the protein is permanently active. That is, the active site is now always open because this protein can't be inhibited, because the inhibitor site is gone, because the protein wasn't made properly. Okay, so depending on where the mutation is in the instructions for making this protein, if the mutation is in the activator site or in the active site, then this protein is permanently shut down and doesn't work in the cells. But if the mutation is in the inhibitor site, then the protein is permanently activated so that it's always active or overactive in the cells. Okay, so mutations, the moral of that story is mutations in different parts of a gene can cause the protein that's made from that gene to either be permanently more active or permanently less active in that line of cells. And that's different than the way the cell normally is, because normally the cell, depending on the environmental conditions in the cell can be more active or less active, but it can always switch between those states. For some of the time when it needs to be more active, it's more active. For other times when it needs to be less active, the cell makes it less active. With the mutations, though, the cell doesn't have a choice anymore. It's either always more active or always less active, depending on where the mutation is. Okay, so that's why it's important to know about normal regulation of proteins in the cell, because first of all, when normal regulatory systems in the cell get messed up, you get cellular diseases. For example, when normal protein breakdown in the cell is messed up, you can get neurological diseases like Alzheimer's. But even when normal post-translational protein sites are messed up, leading to proteins being permanently on or off, you can actually, that can be oncogenic and can contribute to cancer. For example, if a cell reproduction protein is permanently on when it shouldn't be, then the cell will reproduce too quickly and you can get tumors. If a anti-cancer gene, like a DNA repair gene, is mutated so that its active site is messed up and it's permanently inactivated, then that's a problem, because now mutations accumulate much faster in the DNA and the DNA can't be repaired. And so that makes it more likely that the cell will become cancerous. Okay, so I hope at least some of that was comprehensible. I know this is pretty detailed and complex, but as I told you before, living things are the most complicated things we've ever found in the universe. I mean, even the simplest part of a living thing, a cell, requires thousands of different working parts that interact with each other in complicated ways. And this is why it's taken us so long, really, to come up with targeted treatments for cancer. Because just understanding how a normal cell turns into a cancerous cell is super complex. It involves, many different genes in controlling different critical activities of the cell, like cell reproduction and DNA repair. And in some cases, it involves mutating regulatory parts of those proteins to make the protein more active. In some cases, it involves mutating active site parts of the protein to make them less active. And so, this is a really complicated system. So, I mean, if you're having a little trouble conceptualizing, you're not alone. It's complicated, but I'm hoping that as we go through this next part, we talk about targeted therapies for cancer. Knowing this sort of basic biology makes this a little more comprehensible, makes it make a little more sense to you as we go through the more complex parts of these kinds of treatments. Okay, so I'm going to end this video now. I've gone too long on this video. And next time we'll talk about the difference between the targeted therapies and the sort of standard chemotherapies that we've been using, and why targeted therapies, at least theoretically, should be better. And we'll talk about some targeted therapies we've already developed and others that we're in the process of developing. Okay, that's all coming up. So hopefully that'll be a little more interesting to you than this basic stuff was, but still comprehensible, because you're getting a sort of a more detailed background than you would normally get in a bio class. All right, so you take care, and I'll hopefully see you soon.